Ladies and gentlemen, I hope you can hear my voice and also see the presentation on your screen. If you are experiencing technical issues, please let us know through the chat window in the bottom section of the GoToMeeting interface, and we'll try and help you resolve it. Welcome to this LGC webinar on genomic service solutions for plant breeding. My name is Joris Parmentier. I'm the Global Product Manager for LGC Service Laboratories. And well, I'd like to start by really thanking you all for your attendance today. We actually have the highest ever number of registrants for a webinar we've ever organized today. And we've got people from all over the world on the line, from Kenya to all other places in the world. So it's great to see such an interest. Just a couple of housekeeping notes before we get going. Your audio lines are muted, as you might have noticed. That's also because we have so many attendees and we want to make sure that everyone can hear me okay. So if you do have a question, just let me know through the chat message, or alternatively at the end of the session we'll have some room for questions using your microphone as well. If I can get to the next slide. We will cover a couple of very closely related products and services today. We're starting with a short introduction of the company for those of you on the line that are less familiar with LGC, and subsequently we'll discuss the plant sample collection kit, our complete plant genotyping service package, and the turnaround times associated with our service projects. And finally, we are honored actually to also have Mike Olsen from CIMMYT on the line. He'll add a short live contribution with regards to his experiences with the services and products that we discussed today. And as you'll find, Mike has a very impressive track record in molecular breeding, so that's going to be an interesting contribution, I'm sure. And then before the end of the webinar, there's some room for questions. I'm estimating that in total will be around 30 minutes for the webinar, potentially including a couple of questions, might be a bit longer. So short company background to begin with. LGC Group was founded over 170 years ago, I think 173 years ago in 1842, and by now we've got over 2,500 employees working in a variety of divisions, including LGC Standards and LGC Forensics. The Genomics division has service laboratories in London, in Berlin, and in Boston, and additionally we have a representation in all global territories. And from these sites, we support customers all over the world. Over the years, this slide I'm showing now has gotten rather crowded because it's basically showing all the organizations, well, a selection of the organizations we've collaborated with over the years. And it's gotten rather crowded because we work with customers in academia, in government institutes, essentially all the top 10 global ag bio and seed breeding companies, as well as a lot of the smaller ones also. And here we actually only show just a small selection, and specifically the research institutes and government organizations that we've provided our services to. In many cases, we have collaborated with these organizations to actually develop our products and services. Just a couple of examples. We work quite extensively with organizations like the USDA, the United Nations Food and Agriculture Organization, and for example CIMMYT, with whom we've developed a panel of publicly available CASP assays for maize genotyping. To these organizations and many other customers, we offer solutions for DNA extraction, sequencing, and genotyping. Our DNA extraction solutions include several proprietary chemistries and instruments that combine to deliver high-quality DNA. And in terms of sequencing, LGC offers next-generation sequencing as well as Sanger sequencing services. And 2 weeks ago, we actually announced a collaboration with a company called Trade Genetics in Germany. And Trade Genetics is an Illumina CS Pro service provider, as well as an expert when it comes to marker development. And this collaboration will really open up array-based genotyping to our customers, which will make LGC a complete service provider for marker development. And for endpoint genotyping, as many will know, LGC offers instrumentation, reagents, and services using our CASP chemistry and the SNP-line instrumentation. And in a couple of slides, I will show you a short virtual lab tour, but LGC actually runs the largest genotyping service laboratory of its kind in the world. And this really allows us to accept outsourcing projects, even the biggest projects, while still delivering the data in short turnaround time. And these technologies allow us to support our customers throughout their growth as an organization. So whether you're just starting to work on molecular biology or you are already running 100 million data points per year, LGC can really offer solutions to support a wide range of applications as shown here in the slide. Just a couple of examples. Well, we work with a very small company that does not have a genotyping lab, but they do need to run a pre-screening selection on their plants. So in that case, we support them with our all-inclusive service, which I'll come back to, for which they don't need a lab at all. In another example, we work with a company that has been genotyping using our instruments for years. However, this fall, they need to run a 10 million data point marker-assisted selection project, and for which they will not be able to run that in-house. Their capacity is simply insufficient. So in that case, they will actually extract the DNA in-house with their Octopure platform and their SpeedX chemistry, but they will outsource basically the complete genotyping project to us, which will allow them to carry on their day-to-day genotyping in-house. In our service labs, we have experience with samples from hundreds of different organisms, including most commercial field crops, vegetables, fruits, pulse crops, as well as actually human and livestock samples. And the species listed in this slide are actually not all the species we work with, but it's a representation of the more common species. I mean, after all, we're not all interested in DNA extraction or genotyping for racing camels or mini pigs, which we have done in the past. So before we move on to the next part of this webinar, I wanted to give you a very short, let's say, virtual tour of our service laboratory, just in order to give you a bit of an impression of where your samples would be handled if you work with us as an outsourcing partner. So here we see the DNA extraction lab, where we receive various sample types from all the species listed in the previous slide. And we have the capacity to run more than 65,000 DNA extractions per day in our global service labs. In our genotyping laboratories, we have the capacity to run more than 2 million data points per day, and that really ensures that we can cope with basically any project size. And this is achieved by miniaturizing the genotyping reactions into a 1536 well plate, which is shown here on the top left-hand side of your screen, and also by performing the actual PCR with our hydrocycles, which are shown here in the picture. These hydrocycler PCR systems were developed to run up to 64 of these 1536 well plates at a time. And as you can imagine, we're running several of these hydrocyclers at the same time. The run takes about an hour and a half, so we're literally generating not numbers of PCR products, but more kilograms of PCR products. So that's quite a heavy capacity setup. Finally, in our walk-in freezers, we have the capacity to store samples and assays. We do that free of charge, which really allows our customers to quickly and easily perform repeat experiments as well. On our website, we actually have a virtual tour available that allows you to sort of fly through the laboratory in London, and that will really help you get a feel for the scale of the operation. If you're interested, you can go directly to the address shown here, or you can search for lab tour on our website, and I'll also circulate this web address after the webinar. So that's just a little bit on the background of the company. The upcoming 5 minutes or so of the webinar will cover our recently optimized plant sample collection kit. So what is the plant sample collection kit? It's a simple kit that allows you to collect leaf tissue samples for DNA analysis. So how it works is actually pretty straightforward. The kit contains a complete set of materials to collect plant samples in the field, and it includes a leaf punch and a mat, which you can see on the picture here. So after you've received the kit, you start by punching discs out of the leaf with the punch and mat, and you don't have to remove the leaves from the plant for that step. So you can just leave the leaves on. Next, you'll deposit the leaf samples into the provided tubes, and when you've collected all the leaf samples, you close the lids. And when you've collected and when you've closed all the lids, only then you remove the bag of deskant material from its airtight container. That's actually rather important because the deskant material obviously absorbs moisture from the environment, and if it's taken out of the container too early, it'll absorb moisture from, for example, the field that you're working in. And the point, obviously, is that the deskant material is supposed to dehydrate the material in the tubes. So there's little holes in the caps. The deskant material dehydrates the material in the tubes, which prevents a molding, which in turn, and I'll show you a couple of slides on that later, will result in higher quality DNA and subsequently also in higher quality genotyping data. So then finally you seal the package and you return the kits back to LGC for extraction and genotyping. Now we've intentionally kept that kit very small, mobile, and very simple to use as well, just to ensure that you can use it in the field or in a greenhouse, and that it can be used by almost anyone as well. There is a new instructional video available through our website and on YouTube, so if you're interested and if you're planning to work with the kit, it's highly advisable to have a look at that. So why did we develop this kit? Well, firstly, because the use of the kit really improves the quality of genotyping data. And as I mentioned, I'll show you a couple of slides on that. Additionally, it's actually easier to ship samples through customs when they're desiccated. For example, you wouldn't need a phytosanitary certificate. And for those of you who are familiar with shipping biological material across borders, it can cause issues. And this really offers a solution to importing and exporting plant samples from regions with strict customs regulations. Finally, the kit also makes sample handling much more consistent for our lab techs, but more importantly, it also allows us to provide you with better genotyping data. And that's actually the reason why we provide this kit free of charge if you order it in combination with our DNA extraction and genotyping service. So how do we generate better data quality? Well, because the kit ensures that the samples are desiccated, the chance of the samples molding and other forms of degradation are vastly reduced. That in turn ensures that the DNA does not degrade, which allows us to basically more robustly and reproducibly genotype the samples. So here's an example of a genotyping data set from plant leaf material that's collected without using the plant sample collection kits. And these samples are actually extracted using a non-LGC extraction chemistry. We see a call rate of around 71.1%. However, when we collect the samples with the plant sample collection kit and extract the DNA in the LGC service laboratory using the SpeedX chemistry, the genotypes are significantly easier to score, and we can actually get a call rate of 99.5% on the same set of samples. And that really ensures that we are able to generate data for as many markers as possible, which in turn will allow you to identify that marker for the trait that you're looking for faster. For more information, you can visit the PlantKit webpage on lgcgroup.com/plant-kit, and here you can also find some more information on how it works, how you can order it free of charge, and if you order the services associated with it, and also you can find the instructional video there. As a special offer for the attendees in today's webinar, you can also get a free trial kit shipped to your site. You can visit the address shown here on the site, lgcgroup.com/plant-trial-kit, and we'll also circulate this web address so you can easily access a free trial kit shipped to your site after the webinar. So that's it for the plant kit, but if you do have any questions, please don't hesitate to contact me. The next few minutes of the webinar, I'll cover the new all-inclusive plant genotyping service before we move on to the turnaround times, the contribution from Mike Olsen, and a couple of questions. So our complete service really consists of the plant kit, which I've just shown, DNA extraction service, new assay design, and cast genotyping service. And some people within the organization and outside of it actually call it genotyping by courier, because basically all you need to do is you collect the plant samples with kits that we provide, and you send us the samples by courier, and the next thing we'll perform the DNA extraction and the genotyping in our lab, and you'll simply receive your genotyping data by email. The advantages are really that you don't need a lab. The service is also regularly used by customers who do have a lab, but for example, they require additional capacity for a particular project, or for example, when during breeding season, or when they're working with testing sites around the world that don't have a lab in the vicinity. So there's a number of reasons why when this service is very well applicable. Additionally, the plant kit actually ensures the sample quality is optimal, so that you receive better data quality and higher call rate. And for each project, we will assign for you a personal dedicated project manager in the laboratory who can support you throughout the project. And finally, but very importantly, we've priced this all-inclusive service so that it actually allows you to run more samples and more markers. That, in turn, will allow you to identify markers faster, which, in turn, allows you to develop lines with superior production rates quicker and bring these to the market before your competition does. I want to give you a couple of examples on pricing, and actually pricing is also on the web page, so you can find it back there. But here's just an example for the pricing of a parental screening project. The table shows the price per sample, and that's dependent on the number of samples on the left-hand side and the number of SNP markers in the top row. So if in this case the parental screening project has 200 samples and 190 markers to screen, the price per sample would be found by following the green highlights, and in this case you would end up with a price of £25 per sample. So note that that includes that plant sample collection kit, it includes DNA extraction service, it includes genotyping service, all the reagents and consumables that you need, plus that support from your dedicated project manager. But rather importantly, it also includes the design of the new assays. So that means that you're not tied to a fixed panel of assays, and for the next project you can simply swap out some assays for others, or take more, or put more in, or take some out without that changing the price. Note that we also have pricing for up to 500 and even up to 1,000 markers. So that will offer interesting alternatives to the market development work you're doing today. One more pricing example. In this case, for a QC screening project, we're performing with 12,500 samples. These are coming from 25 test fields with 500 samples. And in this case, the price is around £2 per sample, as shown in green. And again, we provide plant kits, new assay design, DNA extraction service, and genotyping for that price. I've also been so bold as to include a small comparison versus Secronome genotyping service. And for the Secronome pricing, we've taken the lowest academic collaborator rate that we can find online, which was about $1,000 per chip and about $20 per assay. And if we then look at the ideal setup really for the Secronome system, which is around 384 samples, and in this case around 48 markers, the all-inclusive LGC price is £6.5 per sample. And with Secronome, we end up with a price of around £8.39 per sample. So that's £1.90 more than with LGC. Another example, 3,000 samples genotyped with 96 markers. With LGC, the all-inclusive service price is £4.50. And with Secronome, because you need to get additional chips and additional assays, we end up with a price of £26.71. That's £22 difference per sample, so almost £67,000 on the total project. Oh, and for Secronome pricing, that is without including the DNA extraction. We've obviously done this for fluorine pricing as well, and the differences are very similar. So that's the all-inclusive service pricing. The pricing is also available online, as I mentioned. For more information, you can visit lgcgenomics.com and navigate to the complete plant service webpage shown here. If you're interested in a project-specific price indication, just let me know after the webinar. I will provide my email address in the last slide. So before moving on to a contribution from our guest speaker, I wanted to spend a very short time on our new service project turnaround times. For our genotyping projects, we offer 2 service levels to choose from. Our express genotyping service with 2 weeks turnaround time from sample receipt to data delivery on the left-hand side of the table, or our regular turnaround time in which we deliver the data in 4 weeks. Interesting to note is that before this summer we actually had a standard turnaround time for all our genotyping projects of 6 to 8 weeks. So without really increasing the price, we have half the turnaround time for all our customers. And finally, our DNA extraction projects, as well as our all-inclusive service, are delivered in approximately 6 weeks. So additionally, bespoke turnaround times are optional. For example, we perform projects in 5 days turnaround time for some customers, and even in 48 and 24-hour turnaround time projects have been performed in the past. So that's about it from my end. Before we move over to some questions, I'd like to hand over the word to Mike Olsen with some real-life examples of how our products and services are used. Mike's been working with molecular breeding in commercial and public companies for many years, as you can see on the slide, and he's quite an impressive track record in molecular breeding. So it's with great pleasure that I can introduce Mike. Mike is dialing in from Nairobi, so I'm hoping we can get a good connection. Are you there already, Mike?
Yeah, I'm here, George. Can you hear me okay?
I can hear you fine. Welcome.
Okay, thanks. Yeah, so I just thought I'd share a little bit of our experience at CIMMYT in the Global Maize program. Over the last year or 18 months, working here out of Kenya, Joris invited me to just share a little bit of our experience. So what we're trying to do is actually to scale up our application of markers in our breeding programs, specifically in response to a recent emerging disease here, maize lethal necrosis, and we have an opportunity to try and bring together a few different disease tolerances into our products. But part of that is actually using a few markers on a large number of samples, and so we've been in discussions with the LGC group, who we've worked with for some period of time, on how to optimize some of these workflows to enable us to look at more plants with a small number of markers. And one of the things that's been key to this successful implementation has been just the forecasting. And I think, from my perspective, just working with the team at LGC, they've been very helpful in that regard, and flexible, and able to accommodate our timelines. So far, again, with these larger projects that we have, the data turnaround time has been really critical. Can really help us reduce our pollinating costs if we can get the data pre-pollination, and with the services we've been able to do that the first few cycles here on a much larger scale in terms of number of samples than what we've run in the past. So again, I just I think that's been really key and really helpful. It involves the forecasting process, involves both parties, of course. I mean, we have to be organized on our end in order for it to work. But when we've been ready and prepared, the LGC group has been really, really helpful from that standpoint. The data quality is quite high. I think we've had no issues at all along those fronts. I mean, we certainly have markers from time to time that are relatively unproven, and it'll be a little bit disappointing when you go into production with them. But the markers, the assays that we're familiar with and that we know work well in the lab, we get excellent data back and the timelines that we need. One thing we've done recently to try and to manage costs on these larger projects is we've been able to aggregate some smaller projects into larger project sizes that enable us to get a little better pricing on the if you look at the pricing options, even on the slide that George sent you can see that there's some economy of scale there as you move towards larger projects. And if we're able to combine projects and run still a fairly small marker set in 5 to 10 markers, something along those lines, and hit all these smaller projects, we've still been able to effectively move ahead and realize some cost savings that way. And I just also thought I'd mention, trying to simplify our protocols a little bit. One thing I would note, when we first started the first cycle with the LGC group, we were running a pretty complex protocol, and the logistics were a little bit challenging. The longer that we've been working together on this, we've tried to simplify our process on our end, and I think that's really made it, in terms of the data turnaround time and everything else, has really improved the overall process. So that's a little bit, I guess, Joris, on our experience on our end. I would mention, certainly, in our situation here in Nairobi, we have access to a small lab. We're able to do a certain amount of DNA extraction, but not on the scale that we're trying to achieve now in a larger breeding applications. So the sampling kits and the courier service, the whole package, has really enabled us to do that. So that's more or less been our experience the last year and a half on working together, and so far we've been really happy with the service.
Okay, great. Thank you very much for the contribution, Mike. For everyone on the line, if you have any questions, now's the time. Questions for myself or for Mike, please let me know, either by the chat window or by raising. If you haven't muted your line, please let us know who you are, what's the organization that you're with, and what your question is, and we'll be happy to see if we can answer that. Are there any questions? Silence remains on the chat and on the line, so I'm assuming that means that there are no further questions. I'll give it another 30 seconds, but if there are no further questions, well, thank you very much for your time and attention. If you do think of any more questions whatsoever, or if you, for example, would like to get a price indication for a specific project, or if you would like any information on anything we've discussed today, please contact me personally. My email address is available on the slide here, so please feel free to contact me. And with that, thank you very much for your attendance, and have a great day.